Thank you for the visit. SciQ is science made simple for the rest of us. Enjoy. We have a symbiotic relationship with the trillions of bacteria that live in our bodies. They help us, and we help them in return. It turns out that they even speak the same language. And new research from the Rockefeller University and the Icahn School of Medicine at Mount Sinai suggests these newly discovered commonalities may open the door to engineered gut flora who can have therapeutically beneficial effects on disease. We call it mimicry, says Sean Brady, director of Rockefeller University's Laboratory of Genetically Encoded Small Molecules, where the research was conducted. The breakthrough is described in a paper published in the journal Nature. In a double-barrel discovery, Brady and co-investigator Lewis Cohen found that gut bacteria and human cells, though different in many ways, speak what is basically the same chemical language based on molecules called ligands. A molecule that binds to another, usually larger, molecule. Building on that, they developed a method to genetically engineer the bacteria to produce molecules that have the potential to treat certain disorders by altering human metabolism. In a test of their system on mice, the introduction of modified gut bacteria led to reduced blood glucose levels and other metabolic changes in the animals. The method involves the lock-and-key relationship of ligands, which bind to receptors on the membranes of human cells to produce specific biological effects. In this case, the bacteria-derived molecules are mimicking human ligands that bind to a class of receptors known as GPCRs, for G-protein-coupled receptors. Many of the GPCRs are implicated in metabolic diseases, Brady says, and are the most common targets of drug therapy. And they're conveniently present in the gastrointestinal tract, where the gut bacteria are also found. If you're going to talk to bacteria, says Brady, you're going to talk to them right there. Gut bacteria are part of the microbiome, the larger community of microbes that exist in and on the human body. In their work, Cohen and Brady engineered gut bacteria to produce specific ligands, N-acetylamides, that bind with a specific human receptor, GPR119, that is known to be involved in the regulation of glucose and appetite, and has previously been a therapeutic target for the treatment of diabetes and obesity. The bacterial ligands they created turned out to be almost identical structurally to the human ligands, says Cohen, an assistant professor of gastroenterology in the Icahn School of Medicine at Mount Sinai. Among the advantages of working with bacteria, says Cohen, who spent 5 years in Brady's lab as part of Rockefeller's Clinical Scholars Program, is that their genes are easier to manipulate than human genes, and much is already known about them. All the genes for all the bacteria inside of us have been sequenced at some point, he says. In past projects, researchers in Brady's lab have mined microbes from soil in search of naturally occurring therapeutic agents. In this instance, Cohen started with human stool samples in his hunt for gut bacteria with DNA he could engineer. When he found them, he cloned them and packaged them inside E. coli bacteria, which is easy to grow. He could then see what molecules the engineered E. coli strains were making. Although they are the product of non-human microorganisms, Brady says it's a mistake to think of the bacterial ligands they create in the lab as foreign. The biggest change in thought in this field over the last 20 years is that our relationship with these bacteria isn't antagonistic, he says. They are a part of our physiology. What we're doing is tapping into the native system and manipulating it to our advantage. This is a first step in what we hope is a larger-scale, functional interrogation of what the molecules derived from microbes can do, Brady says. His plan is to systematically expand and define the chemistry that is being used by the bacteria in our guts to interact with us. Our bellies, it turns out, are full of promise. In conclusion, when we talk to our microbiome, it appears it listens. If we tell it to increase our metabolism and decrease our appetite, maybe it will oblige us. Could this be the start of a new diet fad, called the epigenetic microbiome diet? Only time will tell. If you enjoyed the video, Please like, subscribe, share and leave your comments below. Thanks for watching. Stay tuned for more releases from PsyQ.